Welcome. Let's dive into the fascinating process of human fertilization. Human fertilization is the biological process by which a sperm cell or male gametes fuse with an ovum or female gamete to form a zygote. Zygote is the first cell of new organism. It is a coordinated sequence of molecular and cellular process which begins with the contact of oocytes and sperm and end with the intermingling of parental and paternal and maternal chromosome at the metaphase of the first mitotic division of the zygote. This process marks the beginning of human development. There are some steps in human fertilization. These are: one is ovulation, number 2 sperm journey, number 3 capacitation, number 4 acrosome reaction, 5 fusions of gametes, 6 cortical reaction, 7 formation of zygote, and then zygote undergoes a rapid cell division is called the mitosis. As it moves toward the uterus for implantation, means cleavage begins. Come, let us first learn about the ovum transport. At the time of the ovulation, there is a surge of luteinizing hormone and follicle stimulating hormone. This hormone increase causes the mature Graafian follicle to rupture. As soon as the ovum is released, it is surrounded by follicular cells and some follicular fluid. The follicular cells surrounding the ovum from the corona radiata. After ovulation, the fimbri of the fallopian tube become very active. The movement of the fimbri is hormonally controlled. This finger-like projection helps to pick up the ovum. The fimbri contain mucosal cells that are ciliated and produce a fluid that helps guide the ovum into the fallopian tube. The ovum that is picked up is a secondary oocyte, which is in the process of completing the second meiotic division. If a sperm comes in contact with the secondary oocyte, it triggers the completion of meiosis II, releasing one or more polar body and converting the secondary oocyte into a definitive ovum. Now let's learn about the journey of the sperm. Approximately 200 to 500 million sperms are usually deposited during a single ejaculation. About 2 to 6 millimeters of semen are released into the vagina. 1 millimeter of semen contain more than 10% sperm, while the rest consists of various fluid. In the vagina, sperm exhibit reduced motility due to the acidic environment. However, the uterine cavity provide a more alkaline environment which support sperm motility. Semen has a fruity smell because of the presence of fructose, which serve as an energy source of the sperm. Mitochondria, responsible for energy production, are located in the middle piece of the sperm. It takes approximately 5 to 45 minutes, maximum times 5 minutes, for sperm to travel from cervix to infundibulum of the fallopian tube. Some sperm tubes travel toward the left fallopian tube, while others move toward the right fallopian tube. Semen contain prostaglandin, which help stimulate the contraction of the uterine and fallopian tube muscle during sperm movement. The corona radiata cells surrounding the ovum secrete chemoattractant, which help guide the sperm toward the ovum in the infundibulum. Before actual fertilization can occur, sperm must undergo two essential changes. One is capacitation and other is acrosome reaction. Capacitation in simply a physiological change that increase the sperm ability to fertilize, and acrosome reaction, the release of enzyme from the acrosome to penetrate the ovum's outer layer. Now we start about the capacitation of the sperm. Capacitation is a special type of conditioning that occurs within the female reproductive tract, preparing the sperm for fertilization. The duration of capacitation is approximately 7 hours. During this process, cholesterol is removed from the sperm head by the action of albumin. Albumin is a protein secreted by the uterine glands. The removal of cholesterol increases the permeability of the sperm membrane. As a result, the permeability and motility of the sperm increase, enhancing its ability to reach and fertilize the ovum. Motility of the sperm also increases due to the entry of the calcium divalent cation and HCO3 minus ion into the sperm. This series of changes is collectively known as the capacitation. Let's now understand a few key points about the acrosome reaction. The head of the sperm contains the acrosome, a specialized vesicle. The acrosome contains sperm lysine, including the enzyme hyaluronidase and acrosin. So, the sperm lysine equal to hyaluronidase plus acrosin. These enzymes helps break down the corona radiata, allowing the sperm to reach the zona pellucida of the ovum. The zona pellucida contains a receptor glycoprotein called ZP3, which specifically bind to the sperm head and initiate the acrosome reaction. During the acrosome reaction, the sperm releases its enzyme to digest the zona pellucida, creating a path toward the ovum. Once a sperm successfully fuses with the plasma membrane of the secondary oocyte, sodium ion quickly enter the ovum through protein channel, causing depolarization of the membrane. This rapid change in membrane potential is called the first block to polyspermy. It prevents other sperm from fusing with the oocyte immediately after the first sperm has entered. Remember that the first sperm to reach the zona pellucida may not always be the one that fertilizes the ovum. Only the sperm that successfully undergo the acrosome reaction and fuse with the oocyte membrane can cause fertilization. So, the first block to polyspermy is an electrical response that prevents multiple sperm from fertilizing the same oocyte. It occurs within seconds after the first sperm enter and is temporary, followed later by the slow block, means cortical reaction for longer term protection. Now let's look into the cortical granule reaction. Cortical reaction, in case of cortical reaction, cortical granules are located beneath the plasma membrane of the secondary oocyte, between the zona pellucida and the oocyte membrane. These granules contain peroxidase and GAG. GAG means glycosaminoglycan. Peroxidase increases the cross linkage of the protein in the zona pellucida by clipping protein bonds, making it denser. On the other hand, GAG absorbs water, contributing to the formation of the hard fertilization envelope, a barrier to prevent entry of additional sperm. When a sperm touches the plasma membrane of the secondary oocyte, membrane depolarization occurs. This triggers the release of calcium ion from the endoplasmic reticulum. The calcium divalent cation act on the cortical granules, causing them to fuse with the plasma membrane and release their contents into the space between the zona pellucida and plasma membrane, called the perivitelline space. Keep in mind that calcium ions are already associated with the secondary oocyte, which is arrested at metaphase 2 of meiosis. The entire process is known as the cortical reaction, and it serves as the slow block to polyspermy, ensuring that only one sperm fertilizes the ovum. Meanwhile, the influx of calcium also activates the completion of meiosis II in the secondary oocyte. As a result, the definitive ovum is formed along with the secondary polar body. Finally, the nucleus of the definitive ovum fuses with the nucleus of the sperm, resulting in the formation of a zygote, the first diploid cell of new organism. This is the unicellular embryo. The process of human fertilization is nothing short of a biological masterpiece, from the orchestrated surge of hormone during ovulation to the sperm epic journey through the reproductive tract, and the precise molecular teamwork of capacitation, acrosome reaction, cortical reaction. Every step is a testament to the wonders of. At the end, I would say that the process of fertilization is truly astonishing. Thank you.